Okay, so we will see now the function of the exocrine pancreas. Okay, so the exocrine pancreas secretes a major digestive fluid called the pancreatic juice, which is secreted into the duodenum via the pancreatic duct. Okay, when food is present in the duodenum, the sphincter of OD relax, allowing both the bile and pancreatic secretions to enter into the duodenum. Okay, so what we have seen that the exocrine pancreatic secretes a major digestive fluid called the pancreatic juice, which is secreted into the duodenum via the pancreatic duct. When the food present into the duodenum, the sphincter of OD gets relaxed and allowing the both the bile and the pancreatic secretions to enter into the duodenum by this sphincter of OD. Okay, pancreatic juice. So the pancreas secretes about 1 liter of fluid per day. Okay, the bulk of the fluid is sodium and bicarbonate. The bulk of the fluid is sodium and bicarbonate rich hypertonic alkaline fluid secreted by the pancreatic ductal cell, okay, which together with secretion from the gallbladder and the intestinal juice neutralize acid entering the duodenum from the stomach. Okay, pancreas secretes about 1 liter of the fluid per day. The bulk of the fluid is the sodium and the bicarbonate rich hypertonic alkaline fluid, okay, which uh, contain sodium and bicarbonate with the hypertonic alkaline fluid secreted by the pancreatic ductal cells, which together with the secretion from the gallbladder and the intestinal juice neutralize the acid entering into the duodenum from the stomach. Digestive enzymes. The acinar cells secrete a small volume of fluid rich in the digestive enzyme, the acinar cells. Okay, will secrete the small volume of the fluid rich in the digestive enzymes. These enzymes include the pancreatic amylase, lipase, ribonuclease, and the deoxyribonuclease. Proteolytic enzymes including trypsin, chymotrypsin, elastase, and the carboxypeptide. Okay, proteolytic enzymes include trypsin, chymotrypsin, elastase, and the carboxypeptidase. And there are the amylase, lipase, ribonuclease, and the deoxyribonuclease. Okay, most of the proteolytic enzymes are secreted in the inactive proenzyme. Okay, secreted in the inactive proenzyme proenzyme form to protect the pancreas from the autodigestion and are activated in the duodenum. Okay, most of the proteolytic enzymes will are in the inactive form in the pancreas, and after entering into the duodenum they get activated to protect the autodigestion of the pancreas. Okay, then regulation of the secretion. Pancreatic exocrine secretion is controlled by the parasympathetic stimulation, which enhance secretion of both enzyme and the aqueous component. Parasympathetic stimulation, okay, it's control of the parasympathetic stimulation, then the sympathetic stimulation, which inhibit the pancreatic secretion. Okay, parasympathetic stimulation will enhance the secretion, okay, and the sympathetic will inhibit. Secretin, which stimulates secretion of the alkaline rich fluid from the ductal cells. Secretin will stimulate the secretion. Okay, then the cholecystokinin, which stimulate secretion of the enzyme rich fluid from the acinar cells. Okay, and the somatostatin, cholecystokinin will stimulate, and the somatostatin will inhibit the secretion from both the acinar and the ductal cells. Okay, So we have seen the function of the exocrine pancreas. Then we'll see the function of the large intestine. So the large intestine extends from the ileocecal valve to the anus. Okay, they are extend from the ileocecal valve to the anus. Okay, ileocecal valve. So approximately 1.5 liter of the chyme entered the large intestine per day. Okay, 1.5 liter of chyme will enter the large intestine per day through the ileocecal sphincter. Distension of the terminal ileum results in the opening of the ileocecal sphincter. Okay, distension of the ileum will result in the opening of the ileocecal sphincter, and distension of the cecum causes it to close, allowing regulation of flow into the colon to maximize the function of the large intestine. Okay, and when the cecum will distension, it causes it to close, allowing the regulation of flow into the colon, maximizing its function into the large intestine, which is to concentrate feces by absorbing water and the electrolyte. Okay, in the colon, the electrolyte and water will be absorbed and become, and it makes the concentrated feces. The initial 1.5 liter is reduced to about 150 gram of feces, consisting of 100 ml of water and 50 gram of the solid. Okay, so this many around 1.5 liter of this feces material will then uh, go into the colon, and where the absorption of this uh, water and electrolyte will occur, and this 1.5 liter will convert into the 150 gram of feces. Okay, which consists of 100 ml of water and the 50 gram of the solid. Okay, functional role of the tinea coli. So what is the functional role of the tinea coli? So the muscle layer of the large intestine are slightly different from those found in the rest of the GI tract. Okay, it still has a powerful circular muscle layer, but the longitudinal smooth muscle layer is concentrated into three bands called the tinea coli. Okay, the muscle layer of the large intestine are slightly different from those found in the rest of the GI tract. Okay, muscle layer of the large intestine, okay, are slightly different from those found in the rest of the GI tract. It still has a powerful circular muscle layer, but the longitudinal muscle layer is concentrated into three bands called the tinea coli. These bands are shorter than the circular muscle layer. Okay, these bands are shorter than the circular muscle layer, and gather the cecum and colon into a series of pouch-like fold called the Hostra. Okay, these bands are shorter than the circular muscle layer, and gather the cecum and the colon into the series of pouch-like fold called the Hostra. The cecum and the ascending and the transverse colon are inverted by the parasympathetic branches. Okay, this cecum, ascending and transverse colon are innervated by the parasympathetic branches of the vagus nerve. The descending and the sigmoid colon rectum and sigmoid colon and the rectum and the anal canal are innervated by the parasympathetic branches of the pelvic splanchnic nerve. These parasympathetic fibers innervate intramural plexus, the sympathetic nerve via the superior mesenteric plexus and via the inferior mesenteric and the superior hypogastric plexus innervate the proximal and the distal part of the large intestine respectively. Okay, the sympathetic fibers from the superior mesenteric plexus and via the inferior mesenteric plexus and the superior hypogastric plexus innervate the proximal and the distal part of the large intestine respectively. Handling of chyme. Movement of chyme through the large intestine, okay, involves both mixing via Hustra segment uh, contractions and the propulsion via the peristalsis. Okay, the chyme movement will done in the large intestine involves both the via the Hustra mixing via the Hustra segments contractions, Hustra segment contraction, and the it's called the mixing and the propulsion via the peristalsis. Hustration aid the exposure of chyme to the mucosal surface. Okay, hustration will cause the exposure of chyme to the mucosal surface and helps the reabsorption of the water and the electrolyte. Chyme usually remains in the colon for up to 20 hours. Okay, chyme usually remains in the colon for up to 20 hours. And the parasympathetic stimulation causes the segmental contraction. Okay, parasympathetic stimulation causes the segmental contraction, where stimulation of the sympathetic fiber stops the colonic activity. Parasympathetic will cause the stimulation of the segmental contraction, and the sympathetic fibers cause the stoppage of the colonic activity. The chyme that initially enters the large intestine is isotonic. However, in the colon, more water than electrolyte is absorbed. Okay, chyme that initially entered the large intestine is isotonic. However, in the colon, more water than the electrolyte is absorbed, and the fluid becomes hypertonic, leading to water being absorbed against a concentration gradient. The process of water absorption is controlled by the sodium potassium ATPase located in the basolateral and the lateral membrane of the epithelial cells. The sodium is pumped into extracellular spaces, okay, and the tight junctions at the luminal membrane prevent the diffusion of sodium and chloride back from the extracellular space into the lumen. This leaves a hypertonic solution close to the lumen, causing water to follow by osmosis. The electrolytes are absorbed by a variety of mechanisms similar to those of the small intestine. Okay.